Oh,
This
so
conference will now be recorded.
I was going to say, are you recording it?
Yeah, just started. Okay, and yeah, we do have kind of, I think, a technical issue just with like making sure that we have the information out in advance. So we are going to be taking things kind of slow. And what we thought of is doing another session just to kind of give everybody an overview of the program next time, which would be June, the next Monday. One sec, I'll say, okay, yeah, June 8th. Okay, so let's give this a couple more minutes so that we can have everybody join. In the meantime, what is the plan for today? We're just going to go over the program. What are we trying to achieve in this program? What are we going to cover? And then we can kind of move forward and discuss some of the materials and some of the dates for the upcoming meetings, and start planning how to make this work for everyone. So good morning. My name is Ilya Brodsky. I am from Pine Biotech, and we have been working together with the Louisiana Biomedical Research Network to establish this summer program. The summer program in bioinformatics is going to be focused on working with next-generation sequencing data and utilizing some of the resources that we have put together with LBRN to make this data insightful and informative for your research projects. The program has been open to all of the LBRN, primarily undergraduate institute participants, faculty, and students. And we are excited to have a number of you join us this summer. We are still finalizing the details about registration so that we know that everybody who registered got information on how to join these sessions. And unfortunately, we didn't have too much time to send out this information for this session. So like I mentioned already, we will be recording these sessions, and we will be sending you the recording in a few days when it's complete, as well as sending you the materials. And we will do a repeat of this session in a week from now on July 8th. And hopefully by that time, we will be able to reach everybody and let everybody know about how to join and how to benefit from all of this. So we do have your information, so we know a little bit about you, that you know where, which university you're from, a little bit about your background, why you wanted to join this course, and thank you for providing us with that information. But we will also ask you to share a little bit more about the types of data that you're working with and your experience so far, and how would you be interested in benefiting from working together with us over the next couple of months. So let me start from the very beginning, because I want to make sure that everybody is on the same page about bioinformatics and about what we will be doing. So I will go into a little bit of basics in the beginning, and then hopefully we will quickly transition to the subject matter of this program. Okay, so bioinformatics, as you all know, is an interdisciplinary field that basically applies the principles of statistics and computer science to the domain of biology, meaning to answer biological questions, to work with biological data, and to be able to design experiments, to be able to interpret the data from these experiments, and to make our decisions about biological questions more meaningful. So in that context, I think it's very important to understand that even though all of us can be familiar with all of these aspects of bioinformatics, really we're talking about a team effort most of the time. And so a lot of times in successful bioinformatics projects, you would have people that have deep domain expertise in each one of these areas working together to address one bigger problem. And because of that, I think it's important to all be able to speak the same language. Somebody is going to have maybe more domain expertise in one area of biology where the project is focused. Somebody might have more experience in statistics or utilizing the computational infrastructure that is really needed to address some of these questions. And for some people, that also requires innovation in those computational tools. But ultimately, I think the most successful bioinformatics projects come out of these fruitful collaborations between experts in these different domains. Another reason why it's important to kind of establish a common language between these different domains or areas is because we see the need to really connect these various aspects of how data can impact specifically biology and or even biomedical projects, which is more of a focus in this biomedical research network, the Louisiana Biomedical Research Network, because the data is being generated and it's being generated by technicians or by people that work in labs. And I think understanding how this data is used and understanding what types of data might be informative to address a specific question really helps the technicians make sure that they record the specific metadata that they deal with about the data that they generate, kind of be in communication with the people downstream, and ultimately generate more and better data, which is a critical need in a lot of these projects. Also, I think for bioinformaticians, for people that are joining from a computer science background, I think the motivation there is to understand how the data is generated, why it is generated, what are the specific protocols, and how it's going to be used. And so I think that to improve data analysis, to be able to interpret the data better, it is important to understand the full process. And that's what we're going to try and cover in some of these lectures. I do want to ask for those of you joining to just mute the microphone for now. I will give you a chance to speak as well a little bit later, but for now you should be able to have, yes, the microphone muted. Thank you very much. And finally, I think it's important to understand that some of the data insights that we will generate, the projects that we work on and the insights that we generate from this data is going to be utilized in important decisions. These might be decisions about patients in the long kind of term, but these also will be decisions about specific experiments. And so it's very important for us to understand how this data and how the insights are being utilized and what it takes to get it to that level. So I think that's why this program is important for different types of people that are students and work in labs or plan to work in labs, plan to be in research or even industry at some point. It's important for people with a computational background that will be designing and developing new methods of analysis or combining different methods of analysis to take on larger projects. And it's going to be very important for people that use this insight in making decisions about maybe generating new types of data or setting up a new set of experiments, because they need to kind of be familiar with what methods are there to be able to extract some insights from that data and what is the value of those insights. So the goal of this program is going to be to really start applying the bioinformatics tools and methods to research problems. So what we will try to focus on is making sure that everybody is familiar with how to transfer your sequencing data onto the server where you can run your pipelines, you can do your analysis, different types of data. We'll discuss a few different formats of data. Different formats, of course, have different sizes, and then we have a central repository that we've created for this group, where you'll be able to upload your large data sets, and then we will show you how to start utilizing that data to run some analysis. We will talk about quality control on raw sequencing data, so we'll be able to run some preliminary analysis on the length of reads and the coverage, and understand what these things mean and why they are important for downstream analysis. We'll talk about some pre-processing and filtering, some things that are related to the preparation, the library prep, and the preparation of the data, and understanding how the data was prepared can improve our ability to analyze this data. And we will also talk about just kind of planning, how do we approach different types of questions, so that how do we translate our understanding of what we have to practical steps, and being able to have everything that's needed to execute a successful bioinformatics analysis. In the context of this program, that's going to be focused on next generation sequencing and on RNA-seq specifically, we'll talk about mapping those sequencing reads to the relevant reference genome, how to your genome, how to make sure that it's the right reference. Sometimes we map on the genome, sometimes we'll map on the transcriptome. So we will talk about some of those things. Finally, after it's being mapped, how do we quantify the transcript abundance, and how do we estimate transcript expression from that abundance? Different normalization techniques, and what do they mean? How do they compare between themselves? And also, how do we actually develop or produce a table of expression that has those numbers for each transcript? And then once that table is there, we will use different statistical analysis methods to identify, first of all, differential expression among samples, groups of samples, conditions, treatments. So we'll talk about some aspects of p-values and different testing techniques, and how do we understand the difference, and how do we interpret that difference. But also we'll jump more into the data mining aspect of this analysis. So not just looking at the difference between what I understand my samples to represent, but being able to pull together from different studies, leveraging the online, the public domain resources that are out there, leveraging maybe some of our older experiments and combining them with the newer experiments, and knowing how to operate with these different, more complex data sets. We can use these data mining techniques and extract some meaningful information, and then use it for our next steps. We are assuming that the majority of you already know some of these basic things. So you know what is bioinformatics. You know that there are different methods to analyze many different types of data, not just next generation sequencing, but a lot of other types of biological data. You are familiar with interpretation of that data, how we can leverage online resources and databases, and you already have good experience of posing a question that could be answered with a specific type of data, and you understand how to develop that question based on new data that you have. If you don't, that's okay, and that's even maybe good to kind of refresh our understanding. So really, we've put together a number of resources that you can find on this link on edu.t-bio.info. And so in fact, this program is going to rely on a lot of materials that are provided there. They're covered there in detail as a tutorial, and I'll walk you through today some of the aspects of those materials and the resources that we'll be utilizing. Now, it's going to be useful for several reasons. Obviously, it covers a lot of the things that are written right here. So Introduction to Bioinformatics is a course that will talk about what is bioinformatics and some of the applications of bioinformatics. And then we have a broad overview in a number of types of data that you can analyze, from genomic data to transcriptomic data, metagenomic data, other types of data. And we talk about some of these aspects within the context of a project. So in a lot of these materials, there's going to be a project. And of course, thinking of your own research project is very helpful in the context of other projects that people have been successful with. And so we leverage some of these public domain data sets where you have experts that have published good results and good analysis approaches, and we can kind of build on that to develop a better approach to our own data and our own questions that we would like to use. So in the context of this program, we will have some resources that will hopefully streamline our ability to execute the analysis and plan the analysis, but also to understand the analysis. What are we doing? Why are we doing it? And how to do it better. So one of the aspects are going to be some tools and methods. And so we are going to utilize, you will be able to utilize, of course you're not required to, but you will be able to utilize some of the tools that we provide on the T-Bioinfo platform that allows you to run several different types of analyses. And I think that it will be useful to at least try and understand how it can help streamline that analysis. The second aspect of the resources are these online tutorials that you can always in your own time go and review. They have been put together specifically to address the interests of the people that we have interacted with over the past year or year and a half. And we are going to talk about in these different tutorials online, you will be able to see how some of the same principles of mapping, quality control, statistical analysis, data mining apply to different types of data, and how you can leverage those same types of methods to extract different types of insights from the same data. We also have some prepared project examples. Now, the project examples are in the project section. I will show you in a little bit where you can find them, but also some of these are on different topics, from oncology to infectious diseases to agricultural aspects of different data types. And so what we wanted to put together are some examples that you can kind of build from. Finally, we want to establish a community. So we've been running these programs for some time now. We have a sense of how people kind of go through the materials and eventually put the ideas from these online resources to use in their own projects. And what we have found is that while there is this temptation to kind of jump into doing my own analysis, I already know what I'm doing, I'm just going to run this one pipeline. It's always beneficial to kind of expand, first of all, your horizons and kind of see how other people have done this before, how other people are thinking about this. And so we've spent a substantial amount of time looking at failed pipelines that did not work, or failed experiments that did not produce any interesting biological insights, and trying to think together with people, how do we go back and make sure that we plan it right? And then because we've planned it, we were able to generate some interesting insights into the problems that we were trying to solve. So I think that establishing that community, again, because bioinformatics is such an interdisciplinary field, it's so important to understand who else is in the program, how can we interface with them. And we want to leverage the online, of course, because a lot of these lectures are going to be online, we do want to try and leverage this online community. So let me pause here and just kind of show you where this community is. So you can see that this is the link. I am going to paste it in chat to all of you, and please take a look in your chat and make sure that you can access this page. This is the blog post that we've published about this, and I will just show you how to get, so if you can see my screen down here at the bottom, you can see the organizational pages. And this is going to be the page where you should be able to register for this program. Now, the way to register, if you're not registered on the site, you will see here a price. For you, it's going to be free because you're already registered through LBRN. So just make sure that you have an account. If you are not logged in, right here, you will see a button called register or log in. And so registration is free. You can use your social media account, or you can use your email to register. And once you have registered, please email us that you have registered, and we will add you to this program. So you can email to sahil@pine.bio, or you can email. So what we need is to know that you have registered, and then you can be added to the members of this group, where we will post the materials, and we will also be able to share forums where we post answers to questions or files that we are going through in terms of analysis. So this is somewhere that we would like to interact, but also you will be able to see who else is a part of this program. So for example, Chuma here is from Louisiana Tech, and we have his profile right here, so you can see he was a part of a similar program at Louisiana Tech, and you can kind of see who he is, what he has done, where is he now, etc. So different things like this. So this is just to show you how this works. What else is on here? There's some basic information about the program that you'll be able to see. So some of the upcoming sessions, you can see that we plan to have another program on July 8th, like I mentioned, just to kind of repeat all of the things that we went through today and build on that, because we did just send out the information very recently, and I know that not everybody was able to join. So we did want to make sure that we go through this again. And then we will have another session on July 22nd. I'll go into detail about these sessions, but you can kind of see the upcoming events right here. You can see that we are posting some of the information about today's meeting, for example, how to join, and you'll be able to do that here. We also have here forums that we will be starting to post different answers to questions or any other type of information that we share throughout the course. And you'll be able to see here the progress. So as members of the same group, we will be able to track how people interact with online resources, how they interact with the tools, and that is an important quantitative area for us to measure the activity, address any kinds of issues, and then start to interact one-on-one with each one of you to help you develop a project or kind of review your project and help get to some results at the end of the program that we have scheduled for the second week of August. So this is a little overview. I'll go into more detail again this session a little bit later, and we'll be able to talk about it more in our next session. So just to kind of continue from here, as I said, we'll be using some of these tools and methods that we've provided for this program. We have some online training modules that you will be able to access for free during the program. We have some project examples, and there's this community aspect that you will be able to join. And I pasted the link in the chat. So if you've seen the link and if you have registered during this session, you can also do that. Just let me know. So we will try and add you all even during the session if we can. Now, some of the other things that we'll be talking about is, of course, we'll talk about the T-Bioinfo platform that allows you to process and analyze some of these data sets. We'll talk a little bit about RStudio, how you can build on that analysis and expand it, and maybe leverage some of the public domain tools out there. to visualize your data, to analyze your data. But we'll also talk a lot about public domain data, and I think it's very important for us to be aware of the value that is out there. A lot of these raw data sets that just can inform our analysis, can inform our experiment design, can help us generate a good hypothesis. And I think that there's a lot of these tools, and we'll talk a little bit about how to find data, how to narrow down the thousands of results that you can get for practically any topic that you are planning to study, and how to, for different repositories, they will have different ways of actually accessing the data. So we'll kind of walk you through some of those things. The online training. So I do want to show you here in a second how the online training works so that everybody understands what it is that we actually are referring to by the online training. So as I just showed you, the organizational page that you just saw for the Louisiana Biomedical Research Network Summer Bioinformatics program, on the same site, there are a series of courses on transcriptomics, on RNA-seq, that will particularly focus in this program, but also you're welcome to use some of the other sections, for example, genomics, metagenomics, epigenetics, etc. So some of that data is explained in detail. If you're interested, you can go through those. We'll take a look at that in a second. And a lot of them will have these practical assignments that you can practice and run through a number of project examples, and building that experience, building that skill is going to help kind of translate this into something more impactful in your own research and in your own project. So what is this online platform that I'm talking about? The online platform, you might have been familiar with the platform from other sessions that we did before, or from LBRN. Basically, it's a user-friendly interface for performing these different types of analyses. So all the aspects that we will cover, including quality control, including pre-processing, including mapping, and then statistical analysis like differential gene expression, quantification, also using different normalization techniques, data mining techniques. A lot of that is going to be available here, but it's not a closed platform. It is really a platform that helps you identify best methods and best practices, and then you can read about the individual methods used and utilize those methods in any environment. And so, of course, a lot of these methods are already available on the HPC cluster at the CCT, and also in other places. So this is going to be our training, and you can see here that it covers many different types of data, from next generation sequencing data to mass spec data, structural biology data, and then it has some sections right here on integration, data mining, some utilities for annotation and interpretation of the data, but also some more kind of domain-specific methods. For example, in virology, there's a number of different sections. So the benefit here of the platform is really because it's a known thing. If you just type in bioinformatics headache on Twitter, you'll find a lot of sentiment out there that unfortunately comes with a lot of open source, public domain, great resources. But the problem is that they either just work on the computer that the developer developed them in, or they just work for the data that that team is interested in. And they come up with a lot of interesting ideas and interesting ways of analyzing this next generation sequencing data. However, it's not always straightforward to transfer that from a publication where they publish the code, or some kind of an online server, to incorporate into your own analysis. And so that's one of the things that we wanted to address in this platform. We wanted to standardize how these different tools work so that they work with the same type of input, or it's clear what type of input is needed. They have some settings that you are able to play with and adapt the analysis to your own to your own problem. And they have a consistent interface, right? So at least in a short period of time, you can try out many different things. Now, I also found on Twitter, there's an interesting kind of step-by-step process of how typically one would take a public domain algorithm or a method published and how to kind of walk through using that method. So I have that pasted right here. Right, one, you see a tweet about this new published tool, read the abstract, sounds exactly like what you're looking for. I'm going to just take this data and get a perfect visualization of it. Then first thing, you typically find no link to the source code, but you can Google around and finally you find it. You have to compile and install it. Okay, then you have some library missing, or your version of R is outdated, or some other problem that you haven't accounted for. You have to Google around and spend a lot of time to get it working, run it on some tiny data set that you prepared, get some kind of an error that is very difficult to interpret, right? And then you delete that whole package and never revisit it again. So this is kind of like, I think, an anecdotal, of course, example, but still, I think, true in many different cases. And that's part of the reason why we, first of all, made sure that the tools that we have work in some consistent way, but also in a way that we can interpret. So we understand what is it that we are doing. Now, that's why I think it's also equally important, equally critical for us to understand how to use this data and understand every step of the way. And that's what we want to focus on here. So what we are doing is we're going to be focused on transcriptomics. And so we have a transcriptomics package, so to say, a collection of courses that we hope all of you would benefit from. They start from Transcriptomics 1 and go all the way to Transcriptomics 4. So Transcriptomics 1 is the basics of next generation sequencing. Transcriptomics 4 is single cell RNA-seq. So it goes through 4 different stages to kind of introduce everybody to typical methods in bulk RNA-seq all the way to single cell. And we will utilize those in our program for this summer. So today we are having our kickoff workshop that we will repeat again next week, July 8th, and then we'll be able to send you this information in advance and make sure that everybody's registered, everybody has their updated profile information, everybody has submitted the information that we need to start working on these project ideas. Then we will offer these optional online tutorials that we hope that you will all complete. And every time that you complete this online tutorial, you get points. And so you have to get to a certain number of points to be able to analyze your own data. So the idea is that we've worked with a lot of people that, again, come to the problem of analysis, understanding how they would do it from maybe past experience or what they've read in online literature, etc. And a lot of times some of those things are not accurate and they kind of would benefit from an expanded overview of some of the basics. So those are the things that we put together. And every time that you complete the online material, you get a number of points. Every time that you complete an online assignment, a practical assignment, you also get a number of points. At the end of this process, which we think could be completed in anywhere between 2 and 4 weeks, you get sufficient amount of points to join our transcriptomics workshop. In that workshop, we will work together with you to actually practically build some pipelines, run them, analyze the results, and look at what comes out and how to interpret it. And then you will be prepared both from a theoretical standpoint and a practical experience standpoint to plan your own project. And the session that we will do in that workshop is also trying to see where some of the challenges are coming up. Is it in the planning stage? Is it in collecting the right data? Is it in adapting some kind of a pipeline specifically to the type of problems that you see in your data? Is it just being stuck at some point of that, right? So we will try to push through some of those things. and get everybody started with their own analysis. And then we will have some time until our hackathon. And so the idea behind the hackathon, I mean, we can really view it as a workshop, as a hackathon, it doesn't really matter. What we want to do is we want to make sure that everybody can come together and discuss some of these challenges that they are facing, but not just in the context of I'm facing a challenge and, you know, what would be a way to address it, but let's address it together. Let's get through this, let's solve this problem, let's get it done, let's get to a point where you can talk about your project, your analysis, your results. And that would be the hackathon that is planned for August 9th. We'll do that at the CCT, and we hope that all of you that are going to be working on your own independent projects will be able to come there and kind of bring everything to a close, that you already feel good about it. And then we, like we did last year, for those that have a good project and have made progress, you will have some more time between the end of the program and the annual Bioinformatics conference, where you would be able to submit your abstract and be considered for a poster presentation. And so that is really the plan, is to help you get something out of your own project data, get it analyzed and presented, and get some feedback. And so in between those two, the end of the program and the conference, we'll be able to discuss with you one-on-one or in groups how to make it better and how to make an impactful presentation if that is needed. So again, just to kind of go through some of the dates, today we are in July 1st. So we had originally planned a much longer session to have it between 10 and 1, but we're going to cut it short today because we realized that we only sent the information last night, so not everybody was able to join, and we would really love to go through all of this together once again, make sure that everybody has this information. So we will try and do this again July 8th. So after the session, we'll send out this information again, how to join. We hope that by that time, everybody is already registered on the site and we can see everybody's profiles completed as much as possible. You're of course welcome to, if you're not familiar with a lot of bioinformatics kind of general concepts, please try to go through the Introduction to Bioinformatics module. And then we will start going into the transcriptomics stuff, but also we'll be asking you to submit your project idea, proposal, data, whatever you have, whatever stage you're at with your own project. I know that some of you already have significant data sets assembled, and so we'll start going through, okay, how do we upload this to the server? How do we start the analysis? You know, if you're at some stage of the analysis, how do we review every stage and document that stage so that we know what has been done to the data already? So between July 8th and July 22nd, which is about 2 weeks, we will try to make sure that everybody completes the online modules. Transcriptomics 1, 2, and 3 are really kind of the basic requirement to go through. Once you go through those courses, online courses, plus the information, plus the points that you get from the practical aspect of things, which should get you to about 2,000 points at minimum, right? And so when you get to that 2,000 point mark, that's when you're ready to go into your own independent project. And when you do your independent project, you will get access to a research user on the research platform. That means that you can upload your own data. We'll show you how to upload your own data and start running several analyses and kind of keep track of your analyses to make sure that you understand what those analyses are. Because ultimately what you want to do is complete an analysis, but also have the understanding how to conduct this analysis so that you can reproduce it and you know, replicate it if needed in the future. And then August 9th, which is another couple of weeks after that July session, again, we will have this hackathon, which at that time we will hope to address any issues that are coming up and really making sure that you are not just kind of in the process of analysis, you're really finalizing your analysis. You're at a stage where you're ready to talk about it, and hopefully every team or every person that is participating in this program will be able to share at least something about the projects that they are working on. So at least this is how we are thinking about this right now. Again, you're free to contact us and send us information about where you are in this whole process. And if you need any additional help, we'll be ready to help you there. For those of you at LSU in Baton Rouge, of course, you're all familiar with the CCT, and there's a whole team there that helps maintain some of these sessions. And John, for example, Cubido is with us on the call. So we'll be able to kind of monitor how everybody is doing and making sure, if we know the information, right, if you tell us, we'll be able to address that. So John said in chat right there, hello. So please, I think you'll see contact information kind of popping up in some of our emails. Okay, so again, what will we cover in these sessions? I just want to go a little bit more in detail. But before we do that, I do want to stop talking for a moment, because I have been talking a lot, and make sure that, you know, there's any questions that you might have. Anything you would like to ask or add at this point, you can unmute your microphone and let us know, and that would be very helpful. So again, I'm just pausing really quickly here to let you ask any questions or interrupt me here to say anything that you would like. If you want to just introduce yourself and you know, maybe tell us a little bit about yourself, that's also fine. So you can feel free to unmute your mic.
So I'll just go ahead and say hello to everyone. I'm the computer manager of LBRN. If you all haven't met me, I'm providing support from the LBRN side, but mostly I help people connect together between Ilya and Sahil and our group over here, although Ilya already has a close relationship with our group, so I know that they communicate all the time. So just hello, everyone.
Hello, John. Thank you.
Yeah, a lot of people are shy to speak up remotely. Y'all have a way of communicating via the forums and stuff like that. If they're new to that, that's always a great way to follow up with questions that you think, oh, I should have asked this question when he asked, but I forgot, and whatever. So just encourage everyone to interact with Ilya and Sahil in the forums, if not directly through these sessions.
All right, thank you. And yeah, please don't feel shy. I mean, we are a small group today. We hope that next time everybody will join, and we will have a chance to hear from a lot of you a little bit about your background. I mean, it could be as simple as, hey, my name is Ilya. This is what I am doing right now, and this is what I plan to get out of this program. So again, I'll just give that opportunity again if you would like to say a few words.
Hello.
Yeah, hello.
Hi. I'm Nafe Hyatt. I'm in the 2019 LBRN Summer Research Program. So I've been doing a lot of basic science research for my project involving prostate cancer. So the second phase of my project, I'm hoping to look at some of these computational biology techniques to see how I can further my research. Great,
thank you. And where are you at LSU right now, or where are you located?
So I'm a graduate student at LSUS, and I'm doing research at the Health Science Center, LSU Health Science Center.
Great, thank you. All right, well, we got one person to share, and thank you, Nafai. Sorry if I mispronounced your name there, but we're excited to have everybody here. And again, sorry for the late notice about this event, but hopefully after this little introduction, you get a sense of where we are as a group, and by next time when everybody joins, we'll be able to do this again. So now I just want to quickly go through some of the topics that we will try to cover in these different sessions. Next time, July 8th, after all of us have a great time on July 4th, we will go into more detail with some basics about RNA-seq. And here we'll start talking about how do I find the data, what types of data are there out there that represents RNA-seq or next generation sequencing data in FASTQ or FASTA format. Some of you will be able to maybe find some microarray data. Maybe we can look at TCGA, for example, the Cancer Genome Atlas, where you can find already processed data for, with easy access. So some of those resources, we'll just take a look at what's out there. We'll see how to download that data, where to put it, how big should it be, and when we see the size, how do we kind of understand what the size represents, what type of sequencing, or maybe what stage of processing this would be. And we will see how to upload that data to the server so that you understand the process. Now, we'll also review in detail what is covered in the online Transcriptomics 1 and 2. So Transcriptomics 1 and 2 are these online courses that I'll show you here in a second, where you can find some of the basics about what is next generation sequencing, how to process from raw sequencing files into a table of expression, and then how to work with that table of expression to start infer some information that we can interpret biologically or biomedically. So that will happen in our next session, July 8th. Then later on, we will have a workshop on July 22nd. And in that workshop, we want to be able to go through a lot of the aspects of Transcriptomics 1, 2, and 3, as well as talk about some other sides of transcriptomic data analysis that maybe isn't directly related to mRNA or gene expression. We'll talk about some other types of information we can get out of these files. For example, microRNA or long non-coding RNA expression, or analysis of RNA editing, how you can see alternative splicing, and other types of events and compare them between groups of samples or different conditions. And we'll talk a little bit about how to take the data that we have in the table, maybe some information about ensemble IDs or some kind of reference information, and transform that into something we can understand from a biological standpoint. So those are some of the things that we will cover in the workshop. And then, as I said, on August 9th, you will see an invitation to attend the hackathon. We hope to make this an in-person experience because we think that we'll be able to address everybody's questions and problems quickly together, but it will be also available to join remotely. So obviously you'll be able to kind of see what we're doing, some of us are doing, and see some of the presentations, and also work through your own problems online. So these are some of the events that we are planning. And as I mentioned already, in the meantime, between now and some of these events, we are really hoping that you can join the online environment where we can track your progress and help address any problems, but also so that you are more aware of some of the standard tools, standard processing tools that we have, and start understanding what goes into the processing of this data. So again, I want to go back to this organizational page. So right here at the bottom, you can see the link, and I'll go to this link right now. So I'll transfer here, and just want to show you, first of all, you have to be registered on the platform to gain access here, right? So you should be able to see your account information right here. You can go to your profile and you can see the courses that you have enrolled in. And here you'll be able to see some of the updates that we post and some of the dates of our future events. And of course, you can register in these events. And you'll be able to see the forum topics that we will be addressing, any kinds of questions. I mean, we haven't really posted anything here yet, but we will after this session. So you'll be able to see some of the topics and links and things like that. We have a progress section. Again, I mentioned that everybody has points. So I'll show you in a second how that works. And you can also see the curriculum. So the curriculum will try to address, we'll add this more in detail. What are the topics that we will plan to review in this whole program? Under here, under assignments, we will put practical things that you would benefit from doing. For example, right now, one of the things that we would like to hear from everybody is about your independent project. So the independent project is really the focus of this program. And at different stages, depending on where you are in your own research, you will get a sense of what is the problem statement about what you're trying to solve. So for example, prostate cancer, right? So prostate cancer is a very general topic, right? We can now narrow it down and talk about, okay, so maybe stages, right? So stage, maybe something like mRNA expression changes by stage of prostate cancer. Okay, so I came up with some idea, and now I need to expand on that and say, okay, what am I trying to do? Okay, maybe I'm looking at expression of different types of genes or different transcripts in my data, and I want to understand that change in the context of stage progression of prostate cancer. And then I'll find some raw data that I'll have to find the link for that data, and maybe some reference publications that I've reviewed about this data. If you are working with a PI of your lab, a professor you're working with, somebody else, you can mention that person here. If you generated your own data, you would want to put the experimental person who designed the experiment in here, just so that we have that contact. And if you sequence the data at a lab, you want to just put down, okay, which lab was this? If this is a public domain data set, a lot of this information is going to be in the publication, so it's still nice to have that as a reference. So that would be, you will see different assignments come up here. Under here, you will see the courses that are included in this program, and they're going to be some of these basic courses online. So you can see Introduction to Bioinformatics, Transcriptomics 1, 2, Transcriptomics 3, 4, and then we have Genomics 1 and 2. And of course, some of the other courses are going to be available to you as well. We have other courses that you can see right here, and those include Introduction to Bioinformatics, Machine Learning, Genomics, Epigenomics, Transcriptomics, and Metagenomics. And we are constantly working on additional courses, and also you will have access to some projects. And you can see some of the project examples right here, where if you see something relevant to what you're trying to study, it would be beneficial to kind of go through that. So this is, these are the materials. Again, if you're registered as a part of this program on this link that I just gave, I'm just going to give that to you here again, you will be able to get access to all of this. Okay, so also you will be able to see some of the other members. As I mentioned, a lot of this is going to be useful to you if you're trying to work let's say on a project that is going to be, and sorry, I do hear somebody's microphone being on. So again, if you could just turn it off for now. Okay, so okay, thank you. So about the profiles, so here's an example profile. I already mentioned this before, but it's useful to kind of see where people are. And I mentioned points. The reason why we are putting everything together is because it helps us track where people are and try to address the problems at that stage. So you can see that Chuma, for example, from Louisiana Tech, has a lot of points already, over 9,000 points, and you can actually see some of the types of things that he is doing. And now because we are in the same group, yes, and Utsab, I'm going to address this question about software. So you will see that, for example, he started the pipeline on server tbioinfo, right? And so if you go there, this is actually the account that you will have to request after you register on the EDU site. You will have to request an account and you will get that account. And once you're there, you'll see that you also have an account here. And these two accounts are going to be linked. And they're going to be linked in the sense that you will be able to see your progress on your account over here. And I will show you what that would look like on my account. So for example, as you saw, I have this account, right? And you can see that all of the activity from my account. And sometimes you can see, for example, I worked on some pipeline. So I will see that this pipeline has been finished. And if I click on that link, it will take me to that specific pipeline that I ran. And so it's, again, important, and we'll go through in detail what is all available on this platform in terms of different types of analysis that you can run, how to upload data to be able to run it here, how to download the data, how to download some of the scripts if you would like to modify those scripts further, etc. So this is just to give you an overview again. Okay, so that was the organizational page, and we covered the form that you can use to start submitting your projects that we can consider for the project. So if you don't have your own project, these are some of the projects that we have that you can start with to kind of get a sense of what could be done and where this can go. So we put those in terms of where the data is located, what is the reason we're using specific types of analyses, and what is the output? How do we interpret it and how do we use it? And we'll also kind of work with you to prepare these individual projects that you're working on, whether they're from public domain or they are from your own lab where you generated this, and how to present the data and explain the analysis that you did. So that's going to be a part of what we do. And we've done this before, and we think that a lot of people actually learn from that experience the most, even more than the online materials and workshops like this together. People actually learn a lot when you try to apply these ideas to your own challenges. Like I said, we're going to be able to review how to either enrich the project that you're already working with some of the public domain data. Maybe working with public domain data will give you a sense of where to take your own analysis, what things are possible. And we will give you some examples. So we'll do in our workshop on July 22nd, we'll kind of walk through some of those examples. And here you can see an example of how to find the data to a specific question that you might have. Now, we also want to be sure that we understand how all of you are making progress. And so what we want to collect are pre and post assessment surveys, where we would require a little bit of time from you to read a publication, answer some questions about the publication, so that jointly you for yourself can evaluate how well you understand some of these figures that are typically published in standard publications that utilize this kind of data. And we can understand whether some of the materials that we have provided are helping you get more out of these types of things. So again, what we will use are some publication data, so data sets that have been referenced in a publication. We'll try to find something that kind of fits everybody's interest, whether it's oncology or other projects. We'll share that by next time. And then we'll try and compare the before and after, right? How well do we understand it before the program? How has that changed for after the program? And a lot of times what we see is a lot of improvement, even for faculty and researchers. And I think that plays a huge role in your ability to then independently develop these projects and analyze the data. So again, I am excited about seeing everybody in our next session. That's going to be a little bit longer, and we'll kind of skip a lot of these technical things to jump into a little bit more about RNA-seq and what you can do with this type of data. We'll talk about different types of data that are included in, that vary by the type of method you used to prepare the data. And so these are some of the things that we will cover. We will cover kind of the different data types that can come from next generation sequencing and what are the results of that analysis typically included in RNA-seq. We'll also talk about different file formats, and we'll talk about how we can use the information in these different large data sets like the FASTQ format and the FASTA format and the GTF format, and what other information is needed to process this data and to give us some biologically interpretable result. And we'll also talk about some of the other things that are not just gene expression, right? So we'll talk about the same principles that we use in RNA sequencing. How do they vary between next generation sequencing, like an Illumina sequencer? How do we interpret the same type of information coming from microarrays? How do we use different library preparation techniques like chromatin immunoprecipitation? How do we look at other events like RNA editing, etc.? So we will try and kind of give a broad introduction next time. And then hopefully utilizing the online materials, you'll be able to start generating some kind of a table. And once you have a table, one of the problems is that it's really counterintuitive. So it's not as simple as, okay, so I have these 3 genes and, in this condition and that condition, I can easily understand the variation. A lot of times this data is very complex and counterintuitive, and there's a lot of internal variation that you somehow need to mine for some insights relevant to what you're trying to question. So we are going to talk about some of those data mining approaches as well. And again, I want to kind of conclude this session by asking you to register on the site, start looking at some of these online materials, and especially if you can go, this is the link where you can see the Transcriptomics collection of courses. So you can see Transcriptomics 1, 2, 3, and 4, starting from the very basics of what a lot of that we will cover in our next session, and all the way to single cell, which of course might take a little bit longer time, but still, it might be relevant to some of you. So the link is right here in the chat. You can start looking at that, and hopefully next time we'll have a little more time and everybody will be able to join, and so we'll review some of the stuff, see where people are at, and kind of start talking about some of these projects. So that's all I had for today. I wonder if you have any questions. This would be a good opportunity. You can either add those questions in the chat, and I'll just read through them and be able to answer right now. Or if you have any questions, you can unmute and I'll answer them now. All right, well, in that case, again, I'll just paste this link. So you will be able to actually register just on this link right here. Yep. And then you will be added to to here. Again, if you can email us, then you will be getting added to the program, and we'll be able to keep track from there how to get the most out of this program. So again, thank you everyone for joining today, and I'll look forward to meeting you at our next session, which will be on July 8th. Yeah, so essentially, once you register to this whole organization page, right? So first you have to register to the site, then you will have an account, and then you will be able to register to all of those events. So again, at first when you register and you go here, you will see this price. Once you're a part of this group, it's not going to be there anymore. So you'll be able to register to all the events for free, and of course get access to all of this information, and see that you are a part of this group right here on the activity page. So you can see once I'm registered, you see that it will say that my membership expires September 30th. And then yes, you can register the event to each individual event, or you can register to all events right there. Yes, the price will disappear once you are registered. All right, thank you everyone, and hope to see you again next time.